Oh, thank you. So it's a big pleasure to be here to present in front of this audience, and I'm very sincerely grateful to those brave people that stayed until this moment. So I will be very, very fast, so I will skip over a lot of details. If you have any questions, you can ask later. So just very, very briefly, the hypothesis explaining missing heritability in cancer included common disease variant, which was the basis of GWAS studies. Many common variants of small effect, additive, or pleiotropic effects, gene environment interactions. What I'm interested the most is many rare variants of large effect. Because variants of large effect with even minor root frequency of just 0.5% can explain with a risk that confers risk of 2, can explain 0.6% of sibling relative risk, which is much more than any GWAS hit so far. So rare variants of large effect are almost always founder mutations, and it's probably better to say that founder mutations are always rare variants of large effect. So when we look for rare variants of large effect that can explain significant portion of missing cancer heritability in different populations, which is very important, that can identify these mutations and facilitate genetic counseling or cancer prevention. So, but the problem is, how do we study rare variants of large effect in very large populations? And my solution was a robust and cost-efficient next-generation sequencing strategy that uses pooled DNA samples. And this strategy, I will explain it in a second. This strategy will be very useful for those biorepositories that have been presented before from Greece or India, for example. So the goal was to identify novel, highly penetrant mutations and genes predisposing to hereditary colorectal cancer in this case, using a systematic approach. So we used 1,000 cases, 1,000 controls from Colon Cancer Family Registry core. This is Colon CFR. This is a very large international consortium that has almost 15,000 families with colorectal cancer coming from different sources. And the mean age of cases was 53. The mean age of controls was 66 cases. 66 in controls to adjust for late onset of colorectal cancer. About 90% of them were right. So we selected our cases using different tiers. Tier 1 is FCCTX, is familial colorectal cancer type X, which is hereditary microsatellite stable cancer. Tier 2 is Lynch syndrome, microsatellite unstable hereditary colorectal cancer. Tier 3, 4, and 5, and 6, a different different criteria of hereditary colorectal cancer. So we studied 40 genes. Some of them are established, like ABC or mismatch repair genes, but we also included some candidate and less well-established genes, like LUM. So our strategy started from those 1,000 cases and 1,000 controls, as I mentioned before, that were physically pooled into 3 pools, 3 samples per pool in cases and 8 controls per pool in controls. Then they were sequenced. Targeted sequencing was performed in 40 colorectal cancer genes with the average mean depth of 350x in cases and 100x in controls. So we identified 264 non-silent variants in cases only, not in controls or any other database, and 634 variants in cases and controls. So those that were found in cases only were used for Sanger sequencing analysis. I have to say that we had 0 false positives. So all those variants that we found in next-generation sequencing were found by Sanger, validated by Sanger sequencing. And we used 342 non-silent variants in cases and controls to determine associations. So the outcomes were individual genotypes for very rare variants of large effect. 21% of them were novel. We estimated frequency of common variants and estimated risk. These are not future plans. This is something that is ongoing right now. Segregation analysis in families in collaboration with the University of Melbourne. SNP microarrays for detailed association analysis and functional studies in collaboration with local Alan DeAndre from Harvard. So anyway, the samples, the variants that we identified of large effect were relatively equally distributed between different tiers. As you can see here, so we found about 10 to 15% mutated individuals in each tier. But when we restricted the data to very stringent criteria of American College of Medical Genetics, which almost 100% mutations, the majority were found within the tier 2, which is Lynch syndrome, but there are more mutations in other tiers as well. But this is probably biased because ACMG criteria is heavily based on prior publications. So this is just because of the publication bias, I guess. So we identified 14 mutations, 4 were novel, only 5 mutations were previously classified as pathogenic in ClinVar. There are many more MMR and APC mutations, but we also found new POL-E or NOS1, SH2B3, etc. mutations. In 94 variants that didn't meet the stringent criteria, they still were found in patients with hereditary colorectal cancer and early age of onset, including APC, POL-E1, etc. So some of the identified mutations, this specific gene was found by recent GWAS a year ago. We found this specific mutation in 3 different independent patients with microsatellite stable colorectal cancer, 47, 54, and 62, with multiple relatives with colorectal cancer. NOS1 is from the same GWAS with colorectal cancer, also found 2 mutations, the same mutation in the 2 patients with different early onset of colorectal cancer. Identified new POL-E mutations, very rare case of CHEK2 mutation, truncating mutation in the patient with colorectal cancer. We report for the first time POL-Q, polymerase theta variant that was found in 2 patients with early onset colorectal cancer. And this is the variant that, the variants in POL-Q we're testing right now.
2 minutes.
Yes, thank you. So the association study performed on several, I will skip the technical issues, but these are association hits that we found, associations with colorectal cancer. MSH3 is the most prominent one. The odds ratio is only 1.82, so relatively low. APC that was mentioned by Steve Gruber just recently, just before me, we found 1.6, and I don't know how many Ashkenazi Jews we have, but we do have Los Angeles Hispanics there, so it would be interesting to find out. So the advantages of this pooled NGS strategy that many people were skeptical before we started, but now I have to say it's working perfectly. It's very, very efficient, and it reduces the cost of identifying founder mutations 5 times. So the identification of rare variants without or with very low level of false positives, it's useful for both dominant or recessive inheritance. It can be applied to any condition or with hereditary component, 5 times cheaper as I mentioned before, and it can be used for risk estimation as GWAS for increased GWAS precision, and it can be used for targeted exome or whole genome sequencing equally with equal cost efficiency. I will stop here, and if you have any questions, because the limitations slide is right here, so I can describe limitations personally to each of you. Thank
you. The selected few, the brave, the Marines.
So you have these alleles that they're compatible with the syndrome. So, for example, your MSH3.
Yes.
Right? So, how do you think those rare alleles apply? How do you plan to, how do you think you can incorporate it into risk model, whether it be hereditary risk model or other methods?
I think that they will be incorporated as any other oils. So MSH3 that you mentioned is actually low risk oil. It's 1.8 only odds ratio. Others were found only in cases. So these are mutations with very, very high penetrance. We have to perform segregation analysis first, functional studies, and after that we will decide if they are real mutations.
Right, so they, because one of the things that I'm trying to understand from your discussion is some of these rare alleles, because they segregate in families, they tendency to think that they have the same penetrance as some of the other, high penetrant genes. And until we know where they fit in, the question is how do we use that information? Take choosing plastic example, it comes up, but then, in some populations, it's actually confers some risk of colorectal cancer. So I just wondering how you thought about that.
I think I just did my first step. So what you're asking about is the next one. I definitely agree that this is something that we will need to think about. I was thinking about the discovery first. So we found this alleles. Now it's everybody's turn to study them. And mine also. Thank you.
Good answer.
Great.